Thank you very much for the invitation and giving the opportunity to present our work that we have together with Rabea Madel, who can't attend today, unfortunately, being produced in the lab of Bernd Giebel. So I am presenting our recently pre-printed manuscript, which doesn't work. Now it does work, okay. Independent human mesenchymal stromal cell-derived extracellular vesicle preparations, which differently affect symptoms in an advanced marine graft versus host disease model. And I'm just giving you some background information about MSCs. As you probably all know, these are spindle cell-like shaped cells who have a certain differentiation capacity and do express certain markers as well as known differentiation capacity. And these cells are very prominent in clinicaltrials.gov. If you just search for them, you have about 1,000 registered clinical trials going on for certain disease like diabetes, graft failure, and also GVHD. Certain products have already been launched that are authorized by the authorities and can be used already for GVHD treatment, as an example. They have been used in GVHD treatment since Katharina Leblanc have introduced them in about 2004, where a child was treated with these MSCs from an allogenic source. And we needed to learn 2013 when a negative phase 3 revealed that these cells do not show any benefit by treating these patients. And the question was, why is that so? And we got in contact with our bone marrow transplantation unit. They told us we have a severe ill patient, only 22 years old, and they needed a treatment, and there already MSC-EVs were somehow on the stage to be one of the potential triggering therapeutic agents. And therefore we have implemented their first clinical hands-on study for treating such a treatment refractory GVHD patient. And what we did so far in the beginning, we took cells from the patient and checked whether we could somehow influence the release of cytokines, here pro-inflammatory ones, by giving exosomes in vitro to them. In these times, we called them exosomes. Nowadays, we would call them EVs for sure. And we could see that by applying these EVs, we could reduce the amount of these cytokines, not only in the mononuclear cells, but also on NK cells. We then, because not having any knowledge, because this was a first-in-man trial, we needed to find out the administration dose we could give to these patients. And by that, we somehow made a definition of a certain amount of MSCs secreting these EVs over 48 hours, and then started an escalating dose treating this patient. And during the treatment, we took mononuclear cells from the patient, and what you can appreciate here is by giving these escalating doses, we could again reduce the amount of cytokines in the blood already. We then could, during the treatment, also reduce the amount of diarrhea, here shown as liters per day. And what you can see here is that before the treatment, it was a very high amount of diarrhea that these patients somehow had. And after the treatment, we could reduce this amount. And I think what you can really appreciate is that during the treatment, this patient showed a drastic improvement, not only on her skin, but also on other symptoms. And that gave us somehow the power to go further and reveal, or wanted to bring this product into the clinics. Going into the literature some years after, we found out that these EVs have been used in many in vivo trials already, where they could show not only in the brain or in the kidney, a suppression of inflammation in somehow all of the studies that has been published so far during this period. And this somehow convinced us that these translation of MSC-EVs into the clinics is a good goal, and we went further on that. And Ken already mentioned it, that our group is highly interested in bringing this product there. So we are working together with the ISEF and the ISCT on position papers where our group is taken part for defining what standards are needed and what aspects or quality control criteria needs to be addressed before bringing such a product into the patient surrounding. We also had some common approaches for the COVID-19 statement, because we thought that more and more people are just using MSC-EVs as such and use them in just any non-controlled setting by treating patients. And we somehow reviewed here again that a certain quality is needed and mandatory before you come into the clinical state. So the requirement for the clinical application of MSC-EVs is that the production of these MSC-EVs are with a reproducible activity is given. And this is the goal of the study that I'm presenting today, because what we have done here is we have standardized the procedure for not only generating these EVs, but also in vitro and in vivo testing to see whether such an EV is later on therapeutic active or not. And the first challenge we came across is the heterogeneity of these MSCs that we are used for generating our extracellular vesicles. I mentioned before that we had a negative phase 3 trial for showing that these MSCs might not be the good or a best product to treat these patients. And this might come due to the fact that here showing on the top left side, 20 bone marrow samples being taken from healthy individuals who gave bone marrow for treating patients. So these are healthy individuals giving giving bone marrow, and we raised 20 different MSCs out of them. And what you can just see by eye, that these cells completely differ from each other just by the size, the morphology, and the growth pattern they are having in the cell culture. And if you then go further by taking the conditioned media where extracellular vesicles are released into, these used studies that I showed you before in the in vivo were somehow not being produced the same time. So it doesn't is that easy not only to use these EVs, but the isolation of these EVs have a very heterogeneous procedure behind it, and these are not standardized at all. So the problem is, if you want to bring these into the clinics and everyone does it a little different, you can't compare these results from one to the other. So that is the aim of our study, that how reproducible are these therapeutic effects of independent MSC preparations, and can we predict them somehow in vitro before going in vivo or even into the patients to know that we are treating with the right agents here. So design of the study was the production and functional comparison that we have done from independent MSC EV preparations. And what we have done, we have used 4 different MSCs, all derived from bone marrow, healthy individuals. And one of these MSCs have been prepared in 3 independent batches to see whether we have batch-to-batch variability behind it. These are the results that I can show you today. So what we have done is we have raised them, as I mentioned before, from two clonal cell lines where we have done a well-to-well comparison or just a bulk analysis. And what you can see is that these cells do differ in the growth pattern and in the sizes already. Although by doing the differentiation capability of these cells, we did see that some of them had a very strong capability for doing not only the osteogenic, but also the adipogenic differentiation. Some of them are lacking either the one or the other. So by comparing the flow analysis, by seeing the expression of commonly used MSC markers such as CD44, 73, or the hematopoietic lineage markers, we did see that all of them were expressed, but in a certain extent in a different intensity, for example, seen here in CD73 or in HLA-ABC. So they all fulfilled the ISCT criteria for being MSCs, but they do differ a little. We then took the conditioned media, and what we are doing in our lab is we use human platelet lysate that we produce in our own laboratory. As we are part of the transfusion medicine, we have the possibility to get these platelet lysates for free. And what we do an expansion in 4-layer cell culture flasks. And during the expansion, we can already harvest the media because we do not use any different media for expansion or harvest of the cells. For that, we always use not only the conditioned media, but also just the plain HPL enriched media to produce our EVs. And here we have just recently published our protocol for large-scale isolation. By doing a PEG precipitation approach, we are able to use many liters of supernatant to get an MSC-EV by about out of 8 liters supernatant is our standard volume that we are using here to have many doses later on for comparable side-by-side in vitro and in vivo assays available. Here you can see the independent MSC-EV characteristics that we have done. And what you can see is that the particle number differ between all the EVs that we have done a little, but in size, in protein, and in purity, indicating as particle per milligram protein, they did not differ much from each other. So just by concluding the first conclusion here is that all of our MSCs that we have used for generating our EVs fulfill the ISAT criteria, and all our preparations contain comparable particle numbers, protein, and do not differ much in purity indices. So we then ask ourselves, if they are that similar to each other, do they differ from each other? And here I'm introducing our novel mixed lymphocyte reaction assay that we have established based on a protocol coming from Dirk Strunk's lab, where we are using, if we do from healthy individuals, take the blood and draw the mononuclear cells, plating them into a 96 well plate, wait for 5 days, nothing will happen because these cells will just stay in the culture, they will not proliferate. But if we take mononuclear cells coming from several donors, and we have chosen 12 donors because that is a good amount of cells that we can still handle in the lab, and then pool these cells and then let them incubate over 5 days, you do see that these cells somehow differ from the single ones. So these cells start to see each other as foreign because we have used 12 different donors. So one donor will definitely see another one as a foreign one. And these cells start to proliferate and start to create such patches or clusters of cells. What we then do is we normally take then our EVs for as well 5 days and then incubate them. And what you can appreciate here is that by applying different EV samples into together with the mononuclear cells from these 12 donors, you do see that these EVs have a different influence depending on which EV source we are using here. So we do see that these cells somehow build a different cluster of cells than here. And here we have very high clusters of proliferating cells, we assume. By eye, you can't see what is going on. So that is why we have established a flow cytometry analysis protocol of these. And then just going through this in detail. So what we do is we just harvest the cells and then stain them for several immunomarkers. And then by just defining our cells in the scatter and then discriminating the alive from the dead cells and then going on to the CD4 positive cells, you do see here a very nice population of CD4 plus cells. We here have a stimulation control. So this would be the plain control where we just have the mononuclear cells itself. And you do see here by staining for activation markers of T cells 54 and 25, that you do have a nice double positive population coming up here. If we just add now the platelet lysate-derived EVs from the plain media, you do see that nothing really changes. And if you then apply, in this case, 16.3 EVs to these cells, there is hardly any change in the constellation of these two populations here. If we apply, on the other hand, 41.5 EV population, you do see that this population is massively decreased. And we did observe that in the beginning, and we started to screen with this assay whether our our EVs are capable for being immune modulatory active or non-active, and define them regarding this assay upfront whether they are active or non-active. In the next step, together with Rabea Madel, coming from the lab of Carsten Kirschning, we then have established a mouse model where we have made a transplantation of black 6 mice into bulb C mice. And here we make a T-cell depletion of the bone marrow to be able to titrate the naive T-cells coming from the spleen to have a controlled GVHD induction by the mice. So we transplant these cells into the mice and then wanted to see whether we have any effect. And what you can see is by titrating these naive T cells, doing a clinical score that is harboring by weight loss, posture, activity, fur texture, and skin integrity, that these, sorry, here indicating that radiation only, so this is our control, and giving non-cells, this is down here, that the clinical score is very low. But by increasing cell numbers, you do see, and this is the one that has received the highest number of T cells, these animals need to be sacrificed already after 9 days. Normal time we are observing the animals would be 11 days. So we did find out that something between 500,000 cells and 1 million cells would be the optimal dose to treat these animals to have a high clinical score for seeing a GVHD and being able to suppress the GVHD symptoms by applying our MSC EVs. So, and we didn't just look at the clinical score, but later on, after sacrificing the animals, we did isolate the gut and looked for the integrity of these. And what you can see here is that the gut is already being damaged a lot by using 500,000 cells, but even further, 1 million cells. And you can see here by the histologically scoring that they are very high. So these animals are severely ill. So we decided to not use the 500,000 cells and not the 1 million cells because these animals needed to be sacrificed, but go for 700,000 cells to be transplanted together with the bone marrow. So with this model being established and having a good GVHD symptom behind it, we then ask ourselves which EV fractions would we use to make a proof of concept assay or approach. And being able, seeing that in a stroke model that we are doing together with the group of Dirk Hermann in our university hospital in Essen, we used one that was very therapeutic active in this model. This was 41.5 again. And here what you can see is, so the background of the animal would be that you would give the animal some antibiotics, you do a radiation, you do observe the animals, and then would start the treatment on day 7 for 3 days in a row, and then would wait another 2 days. And after these 2 days, on day 11, you can see that when we apply our EVs to the GVHD mice, you do see a severe improvement of all the morphology in the gut, which is hardly not given anymore. So any structures are not given in the GVHD without any EV treatment. And the histological score of this gut was reduced, severely reduced. And we could also see a positive effect in the PLT count in the blood of these animals. So we did see there is a positive effect by applying our EVs into the animals, which somehow gave us a good start. And then we went on and screened many of our EV samples in the mice, and I will go through you with this a little more in detail. You can highlightly see in green, which is the PBS control. So we implemented a PBS control, seeing here in green, that these mice, after the treatment on day 7, had an increased clinical score, again with the 5 score points I told you before, that these animals are increasingly, the clinical score is increased, and these animals are really suffering. If we apply now here in red our non-active MSC EV preparation 16.3, you do see that this clinical score is even going up to clinical score to 8. And that gave us some concerns, because these animals were really, really sick. Some animals needed to be sacrificed before, because some of our EV preparations seemed to be more non-active than others. So we did see very heterogeneous results here. But if you see in blue now, our 41.5, and here are the ones that we introduced earlier. So we did 3 independent EV preparations on 3 different days. So we raised the cells 3 times independently and harvested the conditioned media and made an EV batch. You do see that these EV batches are able to reduce the clinical score drastically. On this side is the weight change, because this is a very important indicator for doing these observations here. Again, blue is our very active one. So these mice gained in weight again. And because numbers or these schemes are not very good to see what you are doing here, so this is just the mice itself, how they look like. And no GVHD mice would look just as a normal mice. If you treat them just with a PBS, these mice are very poorly. They are doing like a hunching position. They don't move very much. And if you do see a mice treated with a positive or an active MSC-EV preparation, you do see that they are very much active, and this one is treated with a non-active one. So they are very hunching, very poorly. The fur is not very good anymore, and they do start to somehow lose some of the fur as well. Going into further analysis, we did again analysis of the gut, and what you can see here again, that the 41.5, again our active MSC-EV preparation, somehow either rescued or somehow revealed the structure in the gut, whereas the PBS or the non-active EV preparation somehow didn't leave any structure behind. We made a histological score of that again, and as you can see here, the one of 41.5, our active EV preparation, went down drastically, significantly in comparison to the PBS control. We then analyzed further whether we could see any immune function in the gut going on. So we analyzed for the regulatory T cell amount based on FOXP3 staining of the CD4+ positive cells. We did see that both treated mice entities had an increased regulatory T cell amount, but we couldn't observe any differences between the active or the non-active one. And we also wanted to see whether there is any differences regarding organs, and that's why we observed in the spleen whether there is any difference. And what we did see in some animals, there was an increase of the weight, which might be an indicator that more cells are produced and therefore the spleen weight has been increased. We have somehow started to treat the mice with a certain amount of EVs, which came from 2 million cell equivalents. So this was our dose that we gave into the animals. And we asked ourselves whether this was the right dose, and that's why we made a kinetic. So we started to do an escalating treatment of these mice by giving either 1/3, 1 times or 3 times the amount of these EVs to see whether there's any rescue effect or whether any improvement behind it. Here again, indicating the active MSC-EV fractions, and as you can see, that by giving already 2 million cell equivalent EV amount, you do see that there is a reduction, but by giving 3 times the amount, there is no further reduction of the clinical score. So it seems that having an active EV preparation, we can't increase this reduce of symptoms at this time point at least. By having a non-active EV fraction and giving now 3 times the volume of that, we then did see a little reduction of the clinical score, which is not yet significantly, and we have to go into this a little further in the next experimental designs. So having this, we might thought, okay, maybe there's something different between 16.3 and 41.5, which we addressed a little further. And what we did here is we took all the parameters that we have gained in the beginning and looked for it, whether there is any correlation factor behind it. So we did check for the particle number, we did check for the protein, we did check for the purity, we did check for the amount of or the intensity of Western blot, the bands in the Western blots for the protein. We could see, but we couldn't find any correlating factor that would give us a hint whether or not an MSC-EV preparation is somehow having one factor based on the particle number or based on the protein concentration that could give us the therapeutic activity behind it. So to sum that up, we have independent MSC-EV preparations that do differ in their therapeutic activity, and we do also observe batch-to-batch variations that can be seen when applying them more often in the animals. We do think that functional testing of each MSC-EV batch is needs to be tested before any clinical application. And for this, validated in vitro assays are absolutely needed, and they need to be somehow addressed and optimized for the indication that you want to treat your patients of. And surrogate markers, which would be, of course, the beauty that we could somehow see on a cell, whether it will release active or non-active EV samples, would be good, but have not been described yet. And we couldn't find any in our story so far. So this is still ongoing, and we hope to identify them very soon. And with that, I want to acknowledge the team behind it, especially Rabea Madel, who did all the mice experiments, and the team, and of course, you know, all Bernd Giebel, who is our PI and professor behind it. And with that, thank you for your attention.
Well, thank you very much, Verena, for this presentation. We have some people who have already asked some questions in the comment box, and I'd just like to emphasize, if you do want to ask a question or make a comment, please put it in the comment box so that I can call on you and we can have a good discussion here. So I have now, I believe, allowed anybody to unmute themselves, and so I'll just ask for people to please unmute and ask the questions. So we'll start with Thomas Ritter, who has a question about why human MSC-EVs. Thomas, do you want to ask this of Verena?
Yeah, thanks, Kenneth. Yes, so I would be very interested to see what you believe why human MSC-EVs will work in a mouse, because I understand that many people have published this kind of work, but I'm always wondering why human products from human cells, how they would work in animals. So could you comment on that, please?
Yeah, so maybe a little further addiction. So we didn't just check that in the human system, but we also did check that with a mouse system. So we have validated not only a human in vitro assay, but also a mouse in vitro assay. And what we do believe is that on the one hand, we have an immunogenic MSC for sure. And this is why we can also use allogenic transplant in humans, because there are somehow not being recognized by all the immune cells. And what we have to mention here that we are applying in the mouse system, I must calculate, but I think 30-fold more EVs than we would apply in any patient. So the amount we have seen in the patients was that low regarding the body weight that we apply, I think 30 to 100-fold times more into the mouse until we see an effect. And the exact idea behind it is, I think they are immunogenic, and we harbor this as well in the MSC-EVs. So that would be one of the explanations, at least to my understanding.
Okay, but maybe if I add another question here, but do you not think that, microRNAs or whatever is in the EVs, they mediate the effect, and how would they be different from humans compared to mouse?
You mean by applying human MSC-EVs into a mouse system, whether that has any influence here?
Yes.
Well, so we have checked for microRNA, and we did see some differences between the one and the other, but this effect behind it and the mechanism behind it is still under investigation, which I can't really make a comment here because I have absolutely not the idea or the answer yet. Which mechanism is behind it, why an MSC-EV human microRNA would be recognized or not recognized by a mouse.
Okay, thank you.
So our next question is from Yvonne, and her mic is not working. So she asks, I wonder if you just chopped up these MSC-EVs, I guess extruded them, would you see the same effect? That is, does it have to be EVs, natural EVs?
Good question. So we are trying many approaches by changing or influencing somehow, changing the biology of the MSCs to have then also different MSC-EVs. The problem is that we can't really see yet whether if we change anything, for example, by genetic engineering or something, whether this is coming by a different pattern that we see in the EVs or whether that comes by the genetic engineering itself. So we are working here. We have a strategy with Immortalization working on, where we do have the possibility to change also the cells and then change the EVs by somehow harboring different markers to see whether knockout or overexpression will have a different approach here, if that gives you the answer.
Great, thanks for that. Our next question is from Danielle or Daniele.
Yes, thank you for the nice presentation. Did you check the passages or the population doubling times in your population? Maybe I miss it, but do you think that it could be explained, for instance, by replicative senescence or other kinds of senescence?
Yeah. Good question. So what we are doing is we are pooling the conditioned media over several passages. So we have always early and late passages together. At the moment, we are having an approach where we separate the early and the late passage to see whether that has a positive influence or whether the early passages are more active than the old ones. But we haven't tested them yet in the in vivo model. We have just some first ideas in the in vitro assays. But it would definitely have an influence and might be also one of the explanations why the clinical phase 3 has failed, because to my best knowledge, they have used very old cells that have been very often passaged already. And we start our cultivation by passage 6 latest, because then we see that our cells stop somehow being proliferative in a very high population doubling time.
Good. So I actually a very related question to this. I'm going to go to Kui Lin, who has a question now about a marker and how that is related to passage. Go ahead. You want to ask your question about CD19?
Hi, a very nice presentation. So I have this question about CD19 expression and whether you've checked, because in our lab, we actually have some, what I thought was a problem, because as the passage of the MSC gets older, we observe the expression of HRDR and as well as CD19. And it's a little bit strange, and I think that could be related to the senescence of the cells, or there might be an indication of the EV production. And another comment about the therapeutic effect of that is seen in EV. I remember the previous journal club, and there was a talk about comparing the EV and also the concentrated conditioned medium. And then it was found that the concentrated conditioned medium actually works better because there are other secretome in it that is actually required to carry out the immunosuppressive to be more effective. So I have also tried the same experimental design, and in fact, we've set up the mixed lymphocyte reaction, and in this mixed lymphocyte reaction, we've compared the effect of the cell of MSC with that of the extracellular vesicle as well as the concentrated conditioned medium. And we found that in fact the concentrated conditioned medium works best as opposed to just using the EV isolated from the MSC.
Okay, regarding your first question, we don't see major differences regarding early and late passages based on the ISCT criteria marker. So we don't see any CD19 coming up, and we sometimes have a slight expression of HLA-DR, but this is not drastically or very high seen. So this is very randomly seen. So, and regarding your second question, yes, it would be nice to have the conditioned media itself. We tested it and we don't see any effect in our mixed lymphocyte reaction assay. And what we do see is that by applying the empty media without any MSC condition behind it, so the HPL EVs mainly, we don't see any effect. So we always have that as a control to be bypassed with it, to see whether there is anything coming from the media itself. And there must be something that is released by the cells who is the active therapeutic agent or the compartment behind it.
And have you done or have you thought about doing any RNA-seq on the extracellular vesicles to actually fish out what exactly is in the difference of the expression of the of all the genes?
Yeah, so this is a very good question, and believe me, we have done everything that we were able to get access to check non-active to active EV preparations from each other, and we are not able to yet identify one surrogate marker that could explain any potential direction or pathway or functionality behind it. So we are not yet able to see that. We have done microRNA analysis, proteomic analysis. We have done screening of the CVs, seeing for different markers, but we couldn't yet identify one marker. Unfortunately, this would be, as we mentioned before, the gold standard, because that would save us a lot of in vitro and in vivo assays, which are mandatory to our best knowledge.
Okay.
Great. So now a very different question is from Ewan Ross. Ewan?
Hi there. Great talk. Two very quick questions. Firstly, do you take use your donor bone marrow from patients undergoing some kind of surgery, or where are you getting your initial preparations from?
So what we do is these healthy individuals who donate some bone marrow are the ones who go into the clinics to give donation to clinical applications. So they are asked to give bone marrow for a patient, and then they agree on giving us a little extra bone marrow aspirate for research purposes. So they are not treated, they are healthy individuals screened for any infectious markers, so they are absolutely healthy and approved for giving blood for a patient, so they are healthy.
Do you test those preparations for functionality before you start expanding them? We've found that if you test a very early passage and make sure they can still immunosuppress, for example, that gives you a good starting population to then further expand before you do your EV isolations. Obviously, because there's so much heterogeneity in the bone marrow samples that you get.
So as far, we are not testing the cells itself, because what we did observe is that we couldn't define any marker or any test on the cells itself, because we did see batch to batch variations even. So always thawing the same cell give different activities to the MSC-EV. So we do think the culturing itself has a major influence on the activity of the MSC-EVs.
Okay, thank you.
Thanks, Ewan. Costanza, did you have a follow-up? I know we heard from Verena that they threw it at all the omics. Did you want to follow up on the cargo content?
But I just got the one. I think she already replied, but maybe another question will be also to understand if they have a different biodistribution once you put it into the mice. I understand that you cannot find any difference at the level of the cargo, and you did the macroRNA and possibly also mRNA and maybe protein, but try to see if they bind to different cells or with a different affinity.
Yeah, so you probably talk about whether we do see them somewhere in the animals and whether we can see whether they are acting somewhere differently.
Yes, if maybe if they reach different cells or they bind with a different affinity to the same cell type or so on.
Yeah. So all approaches that we have done so far in the lab doesn't give us any good marker or any good staining possibility to track the EVs in the animal. So we are not able yet to track the EVs either during the process while the animal is still alive or after sacrification of the animal. We are working at the moment on a new approach to be able to track the EVs by also genetic modification, but we are not yet there, because what we observed is, and this is based on data coming from a PhD student in our lab, that all the available stainings that are on the market are not specific to EVs, but rather unspecific, and therefore you would never know whether you are really tracking an EV or whether all the byproducts that are in the product, and that's why we didn't put any energy by using these approaches.
But yeah, accepting all the limitation of the current approach, have you tried to see maybe using the old approach if you notice any difference, or you wait for the perfect methods?
Yes, because
to do that.
all,
Okay.
yeah, all the others would not give us a clear answer whether or not it is the EV or whether it is any byproduct that we are tracking there. So that's why the effort behind it is not given to our best knowledge.
Okay, thank you.
Thanks, Costanza. And now we have another question about the source, so the cell source. So Ande Marini.
Yeah, so my name's Andy, and I just kind of wanted to know why you chose like bone marrow derived MSCs as opposed to other sources. And if you think like, do you think you would see the same heterogeneity in other MSC sources, such as from like adipose or hematopoietic stem cells?
Yeah. Good question. Indeed, we are working at the moment also with fetal tissue, like cord blood derived EVs, cord blood derived MSCs, and also placenta derived MSCs. The process behind it, and this is what Ken mentioned in the beginning, the regulatory aspects behind it. So a bone marrow sample is being taken under controlled surgery where everything has been under sterile condition, and all the other sources are under semi-sterile condition being taken. And that's why we have chosen in our hands to go at the moment from the bone marrow derived ones. Another aspect here is you probably have heard about a tissue factor issue, because tissue factor is being expressed on cells coming not from the bone marrow, but on all other sources. And these could influence in a negative manner production of, for example, clots like thrombus in the body. And we probably would like to prevent that. And that is why we have decided to go for the moment, at least with bone marrow. So this is where our production license will go on as well. But we are also investigating whether any cord blood or fetal tissue or adipose tissue would be a possibility at least. But mainly because of regulatory and tissue factor aspects, we are using bone marrow.
Okay, and to kind of follow up with that, so I know some companies such as like Rooster Bio and other companies sell like commercial cell sources from like a single patient that have been like done a lot of studies and like they're commercially available. Would you ever consider using something like that? And if so, what differences would you expect? Do you think there'd be less batch to batch variability between the two?
No, I would not think that any commercial product will have less heterogeneity than ours. I think what the future will bring us is a cell line that we need to use, because we are limited in expansion of our MSCs, so they have just a certain lifetime. And we can't continue to proliferate them endlessly, and that is why we need cell lines in the end. But the question is how to produce them, and again, the regulatory behind this will be very hard to bring them into the clinics, and this is ongoing at the moment as well in our lab, to have a cell line produced that we know produces then all the time active MSC-EVs for a certain disease treatment.
And then my final question is, when you isolated those EVs to use for the various treatments, were they implanted or used in the mice or in vitro studies immediately, or were they stored?
They are all stored. These samples will be stored on -80 in a specific buffer in a validated container that has been put a lot of effort in our lab to define the best storage conditions. And we do see activity remain stable for about 2 years. So when we thaw a sample that has been frozen already for 2 years, we do still see the same activity than we would have done it after 3 months of storage.
Okay, thank you.
Welcome.
Thanks for that. And we have a question, I guess, next from Modeline. Modeline, do you want Modeline Longjohn? Maybe Modeline can't unmute right now, but she asks about EV purity and measures of EV purity. So how do you define purity?
So we do, as every lab, an NTA for particle measurement. We do a protein concentration measurement. We do, because we have the imaging flow cytometry in our lab, we do an analysis of the EVs by flow cytometry. And as I mentioned before, we are using our EVs based on the cell equivalent into the mouse, because we wanted to make sure that the animals can be treated according to their weight. And this is why we apply a certain amount of cell equivalence per body gram weight. And this is what it gives us. But as you did see, the preparations between each other didn't differ that much from each other by using a standardized protocol for the isolation, and the resuspension of the pellet afterwards gives us a very good reproducible product in the end.
So I think maybe this question was answered, but just repeating how you isolated your MSCs and whether you use a commercial source, but I think you cleared that up.
Yeah, so it's a PEG protocol based on homemade or self-made PEG 6000 to reduce the amount of the volume and then go into an ultracentrifugation approach yet. But we are obviously, we want to change that, because that is not a scalable system, because we are limited to 8 liters at the moment in doing 2 UZ runs. The next step will definitely be a scalable closed system for going into the GMP area more. So this will be probably based on either tangential flow filtration or other approaches that we are working on at the moment.
Good. So we have some sleuth work here from Esther Naltatun. Esther, what's your idea here about what might be causing the variability?
Yes, well, if I knew the answer to that question. So thanks, Verena, for sharing these very important data. And I think part of my question you already answered, because you said, well, there's also a lot of heterogeneity in the cells themselves. But I wonder, would it be informative if you say there is batch-to-batch variation in the EV, then also do a parallel analysis of the cells at that time to see whether they also differ in the same way in their therapeutic function as the EV from these cells do, because it may give you some answers on is it really the cell condition that changes and therefore they produce more or less potent cells, or does something else change in this culture, making the isolation different from another batch of the EV? Have you tested this?
So our MSCs are at the moment not in the focus of being tested, because in the end, always our end product is the one that needs to give us the answer whether it is therapeutic,
yes,
active or not.
Of course.
So whatever we do, and if we would identify a marker on the cells that would give us the answer whether it will give us an active or a non-active EV, yes, we would definitely go only for the ones showing or lacking this marker. But as we need to go through the whole process of doing the isolation of our EVs and then having the functional testing, because this will be also mandatory from the authorities that the end product will be tested and not the cell source in the beginning, I think the aim for putting a lot of energy on that would not be the right focus here. So it would be nice to know whether there is any differences, but as we said, the same cell give rise to different EV preparations in the end. We would also need to test always the end product in the end. So that's why we definitely rather focus first on finding a surrogate marker and keeping on testing the EV preparations.
Okay, thanks. Yeah, clear.
So we have a few more questions and a few more minutes here. I want to address, so May asks, have you compared the therapeutic potency between cells, secretomes, and purified EVs? And I know we had, one of our previous questioners had mentioned that in their work, they had compared the secretome versus the EVs and found that in some cases the secretome could be more potent. So there are other factors there. But I understand that here you were just comparing the EVs, correct?
Yes. because for the volume reduction, because it would be too much. So we would need to have any volume reduction before going into the animals, because we didn't see any effect by not doing a purification step upfront in the in vitro assay, to be mentioned.
So, yeah, probably also the part of discussion you have made before, but just wondering if the sample isolation, the preparation process will play the role there as well, and opposed to the secretome and the whole cell in terms of the potency.
So you mean whether the protocol, the PG protocol we are using for doing the isolation has an influence on impact on the activity of the EVs?
Either a different isolation approach.
Okay, well, this needs to be tested for sure. If we do a new isolation procedure, all the quality control needs to be done again, and all the in vitro and in vivo approaches needs to be repeated. We can't just compare one-to-one. But the question is about the scalability, and at the moment it takes us a lot of time to produce these high amounts of EVs, and it would be nice to have a scalable and for automatically procedure behind it, because here we have a lot of open steps, and that will always give us a risk for giving any contamination, for example, and also handling risks. I mean, we have seen that our protocol is very stable and reproducible, but well, yes, new approaches definitely need new validation here. Yeah.
Okay, thank you. Thank you very much.
Great. So, and I think the question from Zach is also similar to what Yvonne asked earlier. If you could just disrupt these, disrupt these membranes, but that's also something that has not been tested in your system yet.
It has been tested in vitro. We have tried to. Well, the problem is you need to first have in vitro a good approach before going into in vivo, because this is a much more work load for the people. And we have tested it, and so far we haven't found any evidence that would bring us into the next step. So we have done a lot of questions here, whether destroying EVs, but how would you define a destroyed EV? And you would need to make sure that this activity then gives us a stable answer. So yes, it's work in progress at the moment, but not to be clear yet which direction it will go. Very
interesting. And then there's a question about immortalizing the populations. I think you mentioned that you're working on that as well. So yeah, we'll look forward to hearing more about how that goes. We've talked already. Amrita asks about the different EV isolation methods. So those are also, like you say, that's a lot of there are a lot of factors there, of course, that could play a role. And then Ana Fernandez Platzkummer asks about the so it's about bioreactors. So the using a controlled bioreactor, and I think we have some people on the call who also have some experience with that.
Yeah, so we have at the moment continued to use a 2D layer stack, so 4-layer stack, because this is the product we are at the moment convinced that it is working, because all our assays that we are doing at the moment, and also the patient, has been treated with a 2D cultured MSC-EV product. But yes, of course, the scalability will only be given when we go into a bioreactor 3D system. And also our group is working on that, but it looks, or it sounds always very easy, but the transfer from 2D and 3D is together with many complications. And this is at the moment our work in progress, where we are trying to define a good, stable process behind it.
All right, 2 more questions. So Ewan has a follow-up here, actually a different question about how you might want to be able to select some specific cells.
Sorry, my head is full of flu at the moment, so my thoughts are a bit random, but have you considered isolating a defined bone marrow MSC population to begin with from your bone marrow samples? So I know it's a bit of a double-edged sword in the MSC field, because some people think you need the heterogeneous mix, but there is subsets within the bone marrow. So have you tried that to see if you get reduced variability within the EV prep?
Yeah, so we have recently seen that a little difference in the raising procedure of MSCs from the bone marrow might have a positive influence on rather getting active EV fractions out of it than the non-active ones. And at the moment, this is being under progress that we are doing it in large scale. So to do reproducible preparations to test them, it might be definitely also one part of the bone marrow taken and then putting the MSCs into culture that might has an influence. Yeah, we are addressing that.
Great, thanks.
Okay, and then Raquel wants to know a little bit more about the storage conditions. Can you tell us more about that? Is that a big secret?
No, I think the big secret behind this is that whatever you do, you need to test and validate the system that you are using. You can't just store them at any condition and tell this is fine. So what we did a screen with about roughly 20 containers with different temperatures and different buffers to see which is the best condition for our EVs being produced with HPL and MSC from bone marrow. And we did see that a special tube from one company gave us the best result with a stable particle count over the time when stored at -80 and the buffer conditions just, so we avoid any PBS, for example. We don't use any PBS in our system. We just use sodium chloride, for example. We do see in the literature that many groups are still working with PBS, and because we think later on for going into the patients, the NaCl or the sodium chloride approach is much more smart, and this is why we use NaCl with a pH buffer and then use them at -80 in this one tube that is in our hands the best, at least.
Good, good. All right. All right, well, I think that brings us to the end of our questions. So Verena, thank you so much for joining us today and sharing this work. I see Bernd is here too. Hi, Bernd. I have to ask one last question though. What is this red thing that's in front of your working group here? Is that a red blood cell?
It is, because we are in the transfusion medicine and we have these ones as like a sitting bench, or like a memorial, like a bench that you can sit in the garden, and they are placed in our university hospital that you can have a rest there. You can sit.
But you're not switching from MSCs to red blood cells just now. Not
yet.
Okay. All right. Well, thanks so much, and thanks everybody for joining today. I really appreciate the contributions and the discussion. If you haven't subscribed to our YouTube channel, we also have a YouTube channel that's called Extracellular Vesicle Club, and we just reached 1,000 followers. So you can join 1,000 of your closest friends and get the updates with the different recordings. So we try to keep all the recordings there. Happy 2021 to everybody, and best of luck with your research and all of your endeavors this year. Hope to talk to you soon. Bye now.